Hello, and welcome to this month's Simon Says Expert Series. To start off, for those of you who aren't familiar with me, I'm Mike Gadus, Advisory Board Member for 100 Voices of Hope at the IU Simon Comprehensive Cancer Center. My mother founded 100 Voices of Hope back in 2008 with one singular goal, and that was finding a cure for metastatic breast cancer. Since inception, 100 Voices of Hope has influenced how innovative research ideas are funded at IU. To date, there's been more than 2.3 million and counting of research funds raised. That has led to 23 bold, out-of-the-box research hunches being funded that otherwise wouldn't have. And those have led to 4 unique clinical trials that can be attributed to 100 Voices of Hope research. Today, I have the pleasure of being joined by Dr. Kathy Miller. She is the Balvay-Lantero Professor of Oncology, as well as being a leader in the Vera Bradley Foundation for Breast Cancer Research at the IU Simon Comprehensive Cancer Center. She's a nationally known and respected for her skills designing and leading breast cancer clinical trials. So with that, Dr. Miller.
Thank you, Michael. It's a pleasure to join you.
You as well.
So we wanted to talk a bit today about immunotherapy and including both vaccines and other types of immunotherapy and how they might impact the treatment of breast cancer. In fact, they already have impacted the treatment of breast cancer. That's been a huge change just in the course of my career. In fact, I've told a couple of people for the first 15 to 18 years of my career, the fact that I never really liked immunology. It was the course in medical school that I struggled the most with. I did okay with the test because I could memorize the things for the test, but I felt like I never really understood the immune system and how it worked. And for the first decade and a half or so of my career, that literally didn't matter. And the first indication, for me at least, that immunotherapy was going to have a role was actually a study where pathologists simply counted the number of lymphocytes, a type of white blood cell, a part of the immune system that had infiltrated a patient's tumor, and showed very nicely that those patients who had a greater proportion of these white blood cells in their tumor at the time of diagnosis had a greater response to chemotherapy, they were less likely to develop metastatic disease, and they had a longer survival. And that really showed us that the patient's own immune system, in its attempts to recognize and control the tumor, clearly had an impact on how that tumor behaved, how effective our therapies were, and how well those patients did. So if the innate immune system has a role, then immunotherapy has to have a role. Now, there's a lot we have to learn about what that role is, how to control it, how to minimize the side effects. But that was a real wake-up call for me that I need to revisit immunology and the immune system and begin to get comfortable working in this field.
Mm-hmm. That's great. Well, thank you for taking the time. Is this the portion where we're going to jump to discussing some of the pre-submitted questions?
Sure.
Okay.
Like, why don't we start there?
Okay.
It may give some focus to my rambling.
All good. And for those of you who might have additional questions, there were questions pre-submitted, but if any additional questions come up, you can put those in the chat box below if you're with us on the web, or if you're with us on Facebook. It's my understanding that you can use the chat thread on Facebook as well, and we'll do our best to get to all the questions before we finish up. So let's start here. Dr. Miller, are there vaccines being developed for various breast cancer types, whether that be triple positive, triple negative?
Absolutely. And I'm going to back up and answer this question broadly first, and then we'll talk about some specific efforts. When we talk about vaccines for treatment of cancer, we talk about it in a very different way than we talk about vaccines for infectious diseases or almost anything else. You don't wait until you have the flu to go get the flu vaccine and think that the vaccine will get you better faster, reduce the severity of the illness. It's something that you take beforehand to try to prevent you from getting the illness or that the course of the illness, if you do come in contact with that virus or that bacteria, will be much less severe. So there are vaccines being developed that are being studied in that preventative strategy, or where we hope they will move to that preventative strategy. That becomes a challenging thing to study because if the endpoint of your study is people who developed cancer, well, you need a huge study because most of the people who enroll in your study who are either exposed to the vaccine or not are not going to get breast cancer. They weren't going to get breast cancer without the vaccine. So there's some real practical and logistical challenges for those sorts of vaccines. But there are absolutely groups working on them. The one that I'm most familiar with is a vaccine that is targeting a protein in the breast called mammoglobin as a normal protein expressed in the glandular parts of breast tissue that gives rise to most breast cancers. And the goal of this vaccine is actually to significantly reduce the glandular breast tissue. So this would be a vaccine potentially, or at least the way we conceive this might be helpful, is something offered to women around the time of menopause or after they have finished childbearing and lactation. Because if this vaccine works in the way we think, lactation wouldn't be possible for women after this vaccine. So it wouldn't necessarily apply to all women. But since 3/4 of breast cancers are diagnosed after menopause, that could still be a substantial reduction.
Yeah.
It's partly because of some of those hurdles that we've looked at vaccines as therapy, either for people who've had cancer and have risk of recurrence to try to reduce their risk of recurrence, or as therapy for people with active disease. Now, I think most people will understand that it's going to be easier for a vaccine to stimulate the patient's own immune system to help fight the cancer when you've got less disease and a healthier patient. So preventing recurrence in people who've had a cancer are getting current best available therapies or have finished those best available therapies, but have a higher risk of recurrence might be an easier task than shrinking gross active disease. We're actually studying vaccines across that entire spectrum. And there have been some vaccines, some developed here by one of my colleagues, Dr. Pravin Kumaya, that have been studied in people with advanced disease, and we are continuing those studies. We have definitely seen improvements in some patients with advanced disease, and we're now moving Dr. Kumaya's vaccines into that earlier setting to try to reduce recurrence. That's both less that you're asking the vaccine to do, but it's also in a patient who we think their immune system will be more able to mount a response to the vaccine and get that protective effect. And those vaccines are different for different types of disease. So some may target the HER2 protein, others may target some of the proteins that tumors use to evade your immune system. Essentially get rid of the tumor's cloaking device. So your immune system is able to recognize them. Some target other proteins that are important for the growth of blood vessels that help feed the tumor or help the tumor grow in different ways. So there are a lot of potential targets where a vaccine could be helpful.
There was one of the pre-submitted questions. Someone wanted to understand, like, can these vaccines detect metastatic breast cancer, or is the vaccine a different method of care than necessarily being able to detect it?
So it's not that the vaccine itself would detect it. And so the way vaccines work in general, and this is true for vaccines that we use for infectious diseases as well as cancer, it is the vaccine stimulates your immune system to recognize whatever the vaccine is against. So if you get the flu vaccine, it stimulates your body to develop antibodies and special immune cells called lymphocytes that recognize the flu virus. So very soon after the flu virus may get to you, your own immune system has already been primed to immediately eliminate the virus so that it doesn't have a chance to make you sick or the illness is much shorter and much less severe. You can think of it almost a little bit of like training wheels. The goal for kids learning to ride a bike is the training wheels are already there before they start to tip, so that they don't tip and they don't fall.
Is is
So in
how
this
is how these vaccines work? Is it the same or different than the other vaccines we're already familiar with for flu, HPV, et cetera?
So different target, but the way they work is very similar. So the flu vaccine is also a protein vaccine. So there are a couple of different ways you could stimulate the immune system to do that preliminary work. We'll call it in the terms of preventative vaccines. One is by exposing the immune system to proteins that are on the surface of the virus that you're trying to prevent. That's exactly what we do with the flu vaccine. The flu vaccine includes proteins that are on the surface of the influenza virus. Your immune system gets educated on recognizing those proteins because you got the vaccine. So if it sees those proteins on the virus, it's ready to jump to attack. The other way of educating the immune system is with what's called mRNA viruses. And that was the technology that was used to create some of the most widely used COVID vaccines. The ones from Pfizer and Moderna were mRNA vaccines. They have the same concept, but at a biochemical level, the way they educate the immune system on what to target is a little bit different. But they all share the same effect. You need to decide what at a very chemical level, what protein are you trying to help the immune system recognize? And then either use that protein itself or the mRNA to educate the immune system to recognize that protein. So it's not that the virus is coding the cancer cells and bringing the immune system in that way.
Okay. Well, realistically, what's the timeline for these types of vaccines to move from clinical trial to being FDA approved and available for high-risk patients in a clinic?
That's always a tough question because it depends on so many factors that are not predictable. Let me tell you the pathway. So we have just launched the first study at IU with one of Dr. Kumaya's vaccines, making that vaccine available to women with HER2 expressing tumors who are at high risk of recurrence. The goal of that first study is partly to look at safety. This vaccine has been used in people before, so we don't expect issues, but that's always important, and even more important when you're looking at moving vaccines earlier in the course of treatment. It's also to look at how well does the vaccine stimulate the patient's own immune system to recognize the targets, and then we can take some of the patient's own immune cells, expose them to breast cancer cells in the laboratory, and make certain that they're actually attacking and killing those cells. So those are the goals of the first study. That first study, everybody gets the vaccine. We're certainly going to follow those women to see if they develop a recurrence of their breast cancer. But without a control and in a study of about 30 women, we're not going to be able to make conclusions on how well this will work. We will be able to say, based on their disease, we predicted that this many women may have had a recurrence at year 3 or by year 5, and it looks like we did better than that, but that certainly won't be conclusive. The next step will be a randomized trial, where Half of the women will receive the vaccine, half won't, so that you really have that direct comparison, that the women in the two groups have similar age, they've received similar treatments, they had similar tumor size, so their prognosis is the same for all the things we know about and can measure. So that the only difference is you got the vaccine or you didn't, to really see if that's successful. That is probably going to be a smaller randomized trial initially to verify that this is still looking very promising. And then we'll need to expand that trial to a larger number of patients to ultimately submit that data to the FDA to get FDA approval. That ultimate trial that will get FDA approval will probably need 1,000 or more women to enroll. So that's going to take some time to get those women enrolled. And because the endpoint is number of women who've had a recurrence of their cancer, we also have to follow them for several years to get those results. So this is not necessarily a quick process, but we don't get to that part if we don't develop the vaccines, test them in the laboratory, move them into patients with advanced disease to look at safety and the response, see activity, and then begin the process. So we're really excited about the study that moves this into people at high risk of recurrence, but without active disease so soon. And we are in the process of writing a trial that moves it even sooner, that takes a different one of Dr. Kumaya's vaccines and looks at adding it to the initial treatment that women receive, rather than waiting until they have finished all of their therapy. So we're committed to moving these things to the clinic as quick as we can, but also trying to accelerate the pace of the clinical research so that if these are successful, they can get to patients as quickly as they can.
For those of us who aren't scientists, could you just clarify, right? Are these vaccines, are they designed to replace traditional treatments like chemo, or are they more of like a booster or enhancer, and we would still expect that those modes of care to still be around?
So it could be a bit of both. So we're currently looking at them in addition to standard therapy rather than instead of standard therapy. And whether they will ever replace standard therapy, I think will be different for different vaccines. So for example, the vaccine that targets HER2 really has as its goal for the patient to make their own HER2 recognizing antibodies. And two of the therapies we give people as an IV infusion right now are antibodies that recognize HER2. So I could imagine getting to a point where we might wonder, instead of giving people infusions, IV infusions of antibodies, whether that component of their therapy is with a vaccine that triggers them to make their own antibodies. Because those engineered antibodies that we deliver as infusions have been such a game changer in treatment, have been so effective, it wouldn't be responsible to look at replacing those at this very early stage. So we're looking at adding to it. But you could envision that being a much later step.
There's someone who is curious to know, would these vaccines in a way help overcome something that could be attributed to genetics?
So they potentially could. As we have learned the function of some of the genes that increase the risk of developing breast cancer, that has definitely led to therapies that are particularly effective for women who have those genetic mutations, and in some cases right now, only effective for women who've had those genetic mutations. So we could also imagine that understanding the function of those genes could point to targets that might only be relevant for people who have that genetic mutation, either in the prevention setting or in the treatment setting.
Okay. There were given for some genetics, and that some might be predisposed based on various factors. Could do you think it'll initially be slowly rolled out for high risk individuals? Or do you think eventually it could be available more broadly to the rest of the population?
So, for a couple of reasons, I think as we're talking about vaccines for prevention of breast cancer in the first place or prevention of recurrence in women who've had an early stage breast cancer, I think the first goals in the first clinical trials will focus on women at high risk. And that will be for a couple of reasons. The practical one is the size of the study that you need to do. I want all of my patients to do really well. But if I'm doing a clinical trial comparing whether treatment A or treatment B reduces recurrence more, if nobody recurs in either group, that's fantastic for all of those women, but it means I can't tell you which is better. So when we design these clinical trials, we have to estimate this is the number of recurrences that I would expect with our current best available therapy in women who have this risk. And I hope that adding this vaccine or this new treatment will make it better by this amount. And then a statistician will tell me how many people I need to enroll in those two groups. So if you have a group at higher risk, the number of people you need to enroll is smaller and you tend to get an answer quicker. The other, though, is with safety. These are still newer ideas. We've learned a lot with some of these vaccines in people with active stage 4 cancer. But both the side effects that are experienced by people who don't have stage 4 cancer and their willingness to accept those potential side effects and toxicities might be very different than in people in the curative setting, many of whom would be fine if I did nothing. And the other is the time course of side effects. If I'm treating somebody with active stage 4 disease, I don't want to hurt anybody with these new therapies. But if I'm treating somebody with active stage 4 disease and you tell me you're worried about side effects that might not become apparent for 10 years, I'm going to struggle not to roll my eyes at you because my physician and scientist brain is saying if this works so well that they are alive 10 years from now to deal with long-term side effects, I'm going to be dancing in the hallways.
Mhm.
That would be fantastic. But in a 32-year-old with stage 2 disease, that 10-year landmark means she's 42. And my goal is for her to be alive at 82 and 92. So those potential rare side effects, but ones that are long lasting or permanent, the late side effects that you don't see until several years after treatment are also a bigger concern. But that means you need to treat people in the early setting and follow them before you can get that information. And that's the other reason why I think those studies will first focus on people at higher risk.
Is it people at higher risk because they've had it before and we're trying to prevent reoccurrence? Or is it going to be a mix of people who've had it, we hope to prevent reoccurrence, or people that might be high risk but have never been formally diagnosed with?
It can be either one. I think the first studies, because of wanting to balance risk and benefit and get answers, will be in women who've had the disease, are at risk of recurrence and preventing recurrence. But one of the things we can look at in those studies is also, did they develop a new breast cancer in remaining breast tissue on either side? Because if you've done it once and you still have breast tissue, you can do it again. That's different than a recurrence elsewhere in the body. But those are things that we can learn from the same studies. In fact, the studies that looked at tamoxifen in women who've never had breast cancer but had a higher risk of developing breast cancer showed that tamoxifen lowered their risk, came from exactly that information. Tamoxifen was first used to treat metastatic disease. It was then used in women with stage 2 and 3 disease who had a risk of recurrence. And we found with tamoxifen, their risk of recurrence was lower. But we also found fewer new primary breast cancers in remaining breast tissue. That's the data that led to those prevention studies.
Fascinating. I have, for right now, I think let's go one more question on vaccines, and then I'm going to flip the topic to topic of the day for a lot of people is AI.
Sure.
Okay. Wrapping up on vaccines, are there specific challenges that have slowed progress on cancer vaccines compared to vaccines for other infectious disease?
Um, I think there are several challenges. One, it is really identifying a target that is important for the cancer, but is not important for healthy cells and healthy parts of the body. That's not necessarily specific to vaccines, but specific to any targeted therapy. If you pick a target that is important for the function of healthy cells in your liver, and you generate a big immune response, it might do great for your cancer, but we might also kill you by all of the liver damage. So one is simply identifying a target that is specific or specific enough to the cancer that it doesn't run into side effects and toxicities. The other is we've learned a lot about how to stimulate the immune system, and stimulating the immune system in a way that is going to be effective. I talked a little bit about lymphocytes, which are part of white blood cells that are part of the immune system. But there are different types of lymphocytes that have different types of different functions. Some of them actually facilitate growth of the cancer and make the environment more welcoming for cancer. I don't want to stimulate those. That's not going to be helpful. So some of it has been the very basic science laboratory work, of understanding the details of the immune function and how those controls work so that we can interact with it in a way that gets the response that we want, that would be helpful. That has come a long way in the last several years, and has really impacted some of the ways some of the current vaccines are constructed.
Okay. All right. So are you ready for some AI curveballs?
Ready.
All right. So let's start maybe up front on some of the diagnosis front. So how would AI maybe help reshape calcification diagnoses early
Oh,
on?
even before calcifications. I can tell you some of the studies that I've seen looking at use of AI in early diagnosis. None of these have yet moved into the clinic, but I actually think they will sooner than some of the vaccine things we've been talking about. So there have been some early studies using AI to interpret mammograms. And AI has been able to identify, I don't know what it is, something in the mammograms of women who were later diagnosed with breast cancer when every expert radiologist has looked at their mammograms and said, normal. We see nothing. No mass, no densities, no calcifications, nothing of concern. Looks good. See you next year. And 4 years later, that woman develops breast cancer. AI looking at those mammograms is able to identify those women in the studies I've seen several years before a diagnosis. Well, if we're thinking about vaccines to reduce recurrence and vaccines in high risk women, imagine the power of having AI read the mammograms and identify women several years earlier. Now, that's not necessarily going to facilitate surgery unless you're talking about prophylactic mastectomy, because the studies haven't been able to identify the cancer is going to develop in the left breast in the upper outer quadrant, 3 centimeters from the nipple. So take this chunk of tissue out. But it could certainly identify women for those early preventative vaccine trials for other interventions that we know could reduce risk. And since AI identified them on mammograms, you could use subsequent AI interpretations to see if you're having an impact on lowering or reducing that risk. So that's one way that it could be helpful.
Okay. So in a scenario where we get to a
Is
day
in
where,
reduce
oh, I'm sorry.
you,
Yeah, I think you cut out for
yeah.
me.
So the other way could be helpful is in helping our radiologists. For even an expert breast radiologist to review mammograms and make certain that they have reviewed them carefully and they have compared to the several years before, it takes a substantial amount of radiologist time. But if you could hand over the initial screening mammograms to AI to read, you could read a lot more in a single day, and that could reduce the reliance on human readers. It could make central reading faster and more efficient, and that could reduce the cost, and that could expand screening on a system level to a much larger population. That may be particularly relevant in lower income parts of the world.
No, I think that's a good segue. Because in a scenario where we achieve a day where through routine screening, AI can maybe detect that I am high risk, right? Or my mother was high risk of developing cancer, but currently cancer free. What then specifically would change about their care plan, right? Am I getting more scans? Am I starting preventative medicine? What does that look like for you?
But there are several things that could look like. I mean, if we focus just on breast cancer, we know that medicines like tamoxifen and raloxifene reduce the risk. They are very much underutilized in high-risk women. But if you had AI tools that could identify women at very high risk even earlier, they may view those options very differently than what they currently view them with their current understanding of their risk. Those are also women, as we're looking at other preventative strategies, would be ideal candidates to enroll in those clinical trials and see if we can develop new preventative techniques. We know that there are some lifestyle factors, having a healthier diet, being at or closer to a healthy body weight, exercising more versus not at all. All of those things are associated with a lower risk of developing breast cancer. That's true in the general population. That has also been true in women who have an inherited genetic mutation, who are at very high risk. Because I think many of our patients want to know, I have this risk. Besides taking pills that I'm maybe not so interested in doing, what can I do? Well, those are things that are in our control that we can do. They don't prevent all cancers, but they certainly reduce the risk. Um, we can talk about enhanced screening. Those may be women for whom adding an MRI to mammography screening it makes sense. Those are women for whom some of the blood tests that are being studied for early detection of cancer might make sense as an augment to their other screening. So those are people who already have options. Um, but that also gives us the possibility of studying other options.
That makes sense. Some of the things we're talking about, whether that be innovations like personalized vaccines, AI driven risk assessments, it can sound a bit expensive to do at scale. So, how do we ensure that these don't become essentially luxury healthcare that only people at major research hospitals can access?
In the short term, some of them probably will. But actually, some of them may decrease costs and expand the people who can benefit. So radiologists are expensive. If I can replace 5 radiologists with one computer, and that one computer can read more mammograms and identify not only cancers that are already there, but women at risk 2, 3, 4 years from now, faster with one computer than I can with 5 radiologists, that could actually reduce costs and expand the population who can benefit. There are similar developmental work going on using AI to read pathology samples, to read the biopsy samples, both for greater accuracy in diagnosis, but also for greater risk prediction. that look very promising. That one computer might be less expensive than the 5 pathologists as well. That could allow people who may have a biopsy in a very rural, under-resourced area to digitally upload the images of that microscope slide to be read centrally, aided by AI. And that may not be very expensive at all to do that. So some of the things that require specialized techniques and detailed molecular testing are very expensive when they first start. But some of the AI tools, I think, very quickly could become ways of expanding access to care, expanding access to these advances more broadly than some other tools have been able to do.
As that occurs, how do you think we'll walk the tightrope of, some might claim, well, AI can detect or see the things that the human eye cannot. So in a scenario where the AI is flagging a concern and that an experienced radiologist isn't seeing, how do you, from a healthcare system perspective, you and your colleagues decide, well, which one do we trust here?
And so it really gets to how do we validate new technologies and take them into the clinic? There are several steps that we and others have thought about for any of these new techniques. So first, it is called analytic validity, which just means if you do the test on Monday and on Tuesday on exactly the same sample, do you get the same result? How reproducible is that result on the same thing? That's often the first step. The second step is called biologic validity. Is it measuring something that we think is important? Whether that's a molecular technique measuring a signaling pathway or an AI technique trying to measure risk. Clinical validity is the next one. Do people who have this thing, whether it's from a blood test or a molecular test on a biopsy or an AI test, is their outcome, is their prognosis different than people who don't have this characteristic or who the test says is negative? Those are pretty straightforward and a pretty clear pathway. The harder one, and the one that you're getting to, is a concept called clinical utility, which is if I, as the physician in charge of this patient's care, manage the care with knowledge of this test or on the basis of this test, does that change the patient's outcome than if I manage their situation without knowledge of that test? That's a much harder question to answer, and it's a bigger hurdle, but it's really the one that we need to bring these technologies into the clinic. If you have a test that just says, hey, Michael, you're at high risk of developing pancreas cancer sometime in the next 3 years, but there is nothing I can do that is going to reduce that risk for you. Well, in the first place, I've just made you terrified, and I didn't change your outcome. In fact, I could argue that I've hurt you because I've given you this terrifying information that has nothing to be done that is going to change that outcome. And that's why we need to think about the implementation and how these tests are used. And that gets tricky because some of that is not about the test. Some of that is about, do I have interventions that are going to change that person's risk?
Yeah.
That's partly dependent on what the test is telling me and how early the test finds it, but it's partly dependent on my preventative or therapeutic options that I have available. So those are also questions that we have to continually revisit, because as either the test gets better or the interventions get better, the answer might change.
Yeah, makes sense. Do you think it helps tailor better, AI will help tailor better treatment plans to either nudge people to do things to help reduce their probability, right? Or maybe if they are being treated, reduce the likelihood of overtreatment to the point where you have negative side effects?
So absolutely. We've talked a lot about AI and these new techniques as ways of adding treatment to people at higher risk. But the other way, better identifying and stratifying people based on risk can be helpful, is by not doing things that we don't need to do. So if the test can accurately identify people whose risk of recurrence is so low, without these toxic therapies, that I don't need to treat them. That's another huge way that they can be beneficial. So I think we'll actually see both. This is there's a Robert Frost poem that comes to mind that when roads diverged in the woods. One goal is identifying those who are at high risk, who are not well served by our current therapies, who need different therapies, new therapies, more therapies. But the other is identifying people for whom we're already doing too many toxic things that they don't need. That's not because we want to, but we've made advances by lumping everybody together, saying everybody with stage 2 disease should get these treatments, because if I do that, I have an extra 2 women out of 100 at my retirement party. And if I don't know who they are, the only way I get treatment to the people who need it is to treat all of them. These tools give us the potential to change that and to focus more and more on people who need treatment, less and less on people who don't.
What do you see as, if we fast forward 10 years, what aspect of breast cancer do you think will look the most different because of vaccines or AI?
Actually think individualizing therapy and maybe not down to the point of truly individual, that this is specifically for Michael Gadus and only for Michael Gadus. But I think that ability to risk stratify, and do more for the people who need it and less for the people who don't. I think that is actually where we will see the greatest impact.
Are there any misconceptions the public has about AI or cancer vaccines that you wish you could correct overnight if you could flip the switch?
Oh, if only I were so powerful. I think that there are always misperceptions about a lot of science. It takes almost always longer than we wish. Lots of things that were really promising didn't work when we first moved them into the clinic, or they didn't work as well as we thought. Um, and I share that frustration. Uh, but in my perspective, that's why the research is so important, because that is the only way that we make progress, and the pace of progress is getting faster and faster. It's also why having teams of researchers that can bring all of these disciplines is helpful. The people who are developing the AI tools need to talk to people like me, not
Mhm.
about their computer code. I can't help them write better code for pulling these features from a pathology slide or from a mammogram. They need me to think about, how might this impact patients? What do we care about? What are the studies we need to do to get this into a responsible use, where we know we're helping people and not hurting them with these tools? Um, that's what makes my role so much fun.
Yeah. And that just the pace of innovation, I know from my mom, she always believed, right, that if she could just hang on long enough, something new was going to pop out, right? Like when back 10 years ago, it was precision genomics, right? She would just
Mhm.
be floored at all this AI stuff and what's possible, even 10 years later.
Yeah,
So
the pace it is so dramatic on multiple fronts, and in ways that we didn't anticipate. And it often coming from very basic research with scientists who are not thinking about clinical application at all. The technology that gave us the mRNA vaccines that led to the COVID vaccines and some of the cancer vaccines that are being studied in treatment came from very basic research trying to understand bacteria's immune system. How does a bacteria protect itself from a virus that is trying to infect that bacteria? That was the question that researchers were studying that gave us the technology that ultimately led to mRNA vaccines. So the investment in clinical trials, I think, is easier for people to see the human impact
Mhm.
of that and how that may affect real patients. I think it's harder for people to see how investment in somebody studying bacteria and viruses can really impact people. But that really fundamental, basic understanding provides the crucial building blocks that get us there, and it can get us to places that were not on anybody's radar when they started that research. And that's part of what I find so fascinating.
Yeah, there's a component of it that almost has a butterfly effect element.
It absolutely does.
Yeah. Well, I thank you so much for your time, Dr. Miller. This has been a great discussion, and thank all of you who joined us on the web for this month's Simon Says. There will be a recording of today's session and information about upcoming sessions that you can get from cancer.iu.edu/Simon Says. Take care.
Thank you, Michael.